Welcome to the last topic in Chapter 2 on molecular biology, Chapter 2.9 on photosynthesis. Photosynthetic organisms are always at the beginning of every food chain, okay? And because of that, they're often called primary producers. Okay, now it's really easy to say that plants are primary producers, but plants aren't the only things that can undergo photosynthesis. So some other examples include things like algae. Some bacteria can undergo photosynthesis, okay? Things like phytoplankton. Okay, so even though we're going to talk about plants and mention plants, photosynthesis really applies to a whole bunch of other organisms. Let's start with the overall equation for photosynthesis. So plants and other things are going to take carbon dioxide and water, and through the magic of photosynthesis, they're going to turn that into glucose, C6H12O6, plus oxygen. Now, some of you are probably tempted to write light over here, and you may even see that in some spots, but light doesn't have mass, so we generally don't write it here. If you're dying to write it, you can write it over the arrow. Okay, and when you do that, what you're saying is that this kind of happens, okay, this reaction happens with the help of light. So let's talk about energy here. Okay, so my energy is coming from light. Okay, that's where the energy is coming from. So we go from light energy. Okay, and if I'm looking at these products, the product that has most of the energy is this glucose molecule here. And that is in its bonds. And bond energy is better known as chemical energy. Okay, so I kind of like to write it like this. Okay, during photosynthesis, we turn light energy into chemical energy in the bonds of glucose. Okay, so we just said that photosynthesis needs light. Well, visible light is only a very small part of what we call the electromagnetic spectrum. Okay, and the electromagnetic spectrum, as you're probably already familiar, comes in waves, and these waves range in wavelength, okay? So in terms of wavelength, okay, we're going from small to big, okay? So we can see that these waves have really small spaces between them. These waves have really large spaces between them. Let's talk about energy, okay? So these waves have energy. The faster or more frequent these waves are coming, okay, the more energy they have. So energy is actually going to work the other way. So things with small wavelengths have large amounts of energy. Things with large wavelengths have relatively small amounts of energy. All right, so visible light, okay, is somewhere in here. It's kind of like somewhere in the middle, and it's only going to represent a relatively small portion of wavelengths. Okay, as we kind of go towards the smaller wavelength end, okay, next thing is going to be UV light, followed by X-rays, and gamma rays. These are some really gnarly things, okay? It can cause quite a lot of damage to cells and such. Okay, but in terms of energy, visible light has less energy than UV light, X-rays, etc. None of these are visible. The only visible ones are in here. All right, towards the larger wavelength end, we're going to find infrared and radio waves. Okay, so out here, less energy. Again, as humans, we can't see these, so I should kind of clarify that. When I say visible light, I mean visible to humans. Different organisms can perceive different wavelengths or ranges of wavelengths. So I'm going to kind of limit this to what we call human visible light. Okay, so if you're drawing the spectrum for visible light, so this is only what is visible. We just took a chunk out of this crazy thing, okay, and we're just looking at one section of it. If you're drawing this, it's probably not being drawn in color. So I would just say down here are my blues. In the middle, I have my greens. And up here, I have my reds. Okay, so the wavelengths look something like this. These are a lot more energy. And then as the wavelengths, wow, that's really bad. As the wavelengths get bigger, okay, and these are wavelengths, bigger wavelengths, the energy is actually decreasing a little bit. Okay, so let's talk about white light for a second. White light contains all of the colors of the visible spectrum, okay, and when light strikes an object, one of two things are going to happen. Either the object absorbs that light or the object reflects that light. So let's think about why something like this ladybug might look red. That must mean that it's absorbing all of the colors and reflecting the red light, and that's why it looks red, okay? Let's take a look at what colors plants are able to absorb, okay, and which ones they reflect. So take a moment to kind of draw this out. On the x-axis, we have wavelengths going from 400 to 700, so we're in that visible spectrum. Down here are going to be our blues, our greens, and our reds. And up here we have percent absorption. Okay, so you guys are no dummies. You know that chlorophyll appears to be green. All right, well, again, it appears to be green because down here in the green ranges, we're getting a relatively low, okay, absorption from chlorophyll of those green lights. That must mean that it's absorbing the other colors, like the blues and the reds. Okay, so realistically, the general trend for light absorption for chlorophyll looks something like this. Okay, really high up here, low down here. In reality, it looks something like this. Okay, some kind of crazy shape. You don't need to know that, okay? The trend that you need to know is that it absorbs it absorbs blues and reds really well, greens not so much. Okay, so this question you should be able to answer on your own, okay? Plants look green because their main pigment, which is chlorophyll, absorbs all of the wavelengths except green. Green gets reflected to your eyeballs, and that's why your brain perceives the plant is looking green. All right, before we go on to label a chloroplast, okay, let's talk about plant cells in general. So let's see how much you remember. This big empty space here isn't an empty space at all. That's the central vacuole, probably filled with water. All of these things that are kind of lining the periphery, and I see a bunch of them here. Whee! Well, maybe not that last one, okay? These are all chloroplasts. So this is kind of what they look like in scanning electron microscopy. And this, I would venture to say, is the nucleus. Okay, I can see some chromatin material spread out through here. All right, so what we're going to do is we're going to attempt to draw one of these suckers. And I'm going to draw it like this. And just like the mitochondria that we've discussed earlier, we think that chloroplasts were once engulfed by early eukaryotes in what we call the endosymbiotic theory. okay? And these suckers already had their own membrane, but when they got engulfed, that added another membrane, okay? So our chloroplasts actually have two membranes, an inner and an outer membrane. So let's label those now. So outer membrane, inner membrane. Okay, so unlike the mitochondria, the inner membrane kind of just sits right inside of the outer membrane. It's not all squishy like. Okay, now let's talk about these dark stacks of things here in these chloroplasts. Those are what we call thylakoids. Okay, so a single disc is called a thylakoid or thylakoid disc. An entire stack of them is called a granum. Okay, grana would be plural. And I'm not going to draw a bunch of them. So here's a granum, here's a granum, here's one, here's one, here's one, here's one, here's one. So obviously they have multiple grana in each chloroplast, but then my drawing would be a mess. So I'm just going to draw one. And then we can also see here that a lot of the chloroplast is filled not with grana, okay, but kind of like a jelly type substance called the stroma. So the stroma is just kind of what fills up the chloroplast, not included in the thylakoid. And it actually has mass. It's like a jelly water-based substance. Okay, so just some general things we should know before we move on. Here's an SEM image of a chloroplast, and again, we can see the thylakoid discs here and the stacks of them, which are the grana. And then these kind of spaces in between are filled with the stroma. So we should expect chloroplasts to be green, but not the entire chloroplast. Only the thylakoids are green because that's where the chlorophyll is. We know that chlorophyll's role is to absorb light energy. So if something doesn't have chlorophyll, like the stroma, that should tell us that it doesn't absorb light. And that's really important because photosynthesis is going to occur in two stages. One that requires light, and if that's the case, I better find chlorophyll, and one that does not absorb light. Now, one of the first things that's going to happen in photosynthesis is this process called photolysis. Okay, we've talked about a lot of words that end or include the word lyse, okay? Hydrolysis, cell lysis, the lytic cycle, glycolysis, and that literally means to break. So we know lysis has something to do with breaking or splitting, okay? Photo means light. I will tell you, we are not breaking light. That's not how it works. Just like in hydrolysis, where we're using water to break something, here we're going to use light to break something. And that something happens to be a water molecule. So in photolysis, we are going to split a water molecule. So here's my Mickey Mouse looking water, and I'm going to be splitting it using the energy from light. That, of course, is going to happen in the thylakoid. So here's a thylakoid, and I know it has to be a thylakoid because thylakoids contain chlorophyll, and chlorophyll is the pigment that actually absorbs the light. Okay, so let's see if we can fit the entire process of photosynthesis on one tiny page. Okay, so I know photosynthesis occurs in the chloroplast, so I'm going to go ahead and draw and maybe label a chloroplast. And in this case, I'm just going to draw one thylakoid. Holy crap, that says thylakoid. Thanks for being patient, okay? Because I don't want to muddy up my drawing with a bunch of other things, okay? And this space over here, of course, is called the stroma. Okay, so we said one of the first things to happen was photolysis. And for photolysis, I need two things. I need a water molecule, okay, which I'm going to send to the thylakoids. And I need light energy, okay, probably from the sun, but it could be from a lamp. Okay, now we're going to use the energy from light to do two things. One of them is photolysis. The other one we'll get to later. So when we split this water molecule, we're going to split it into H's and O's, okay, hydrogens and oxygens. The oxygen is not needed, okay, so we are going to get rid of that as a byproduct. Okay, and now I'm left with these hydrogens. Okay, these hydrogens need to somehow make it over here, okay, for the next process, but they can't go on their own. Okay, we're going to send in a carrier molecule, and that carrier molecule is going to carry these hydrogens as well as some of the electrons that were released from this process. Okay, so we're going to send in a carrier called NADP. Okay, NADP is going to come to the thylakoid. It's going to pick up an electron, which we don't really write, and hydrogen, and it is going to become NADPH. Okay, so that H is coming from here. Okay, so we said that we were going to use the energy from light to do two things. One was photolysis, which we just did and we just finished up. Okay, and the other thing that we're going to use that energy for is to recharge a DP into a TP. Now, because we need light for all of these things, we call this the light-dependent reaction. Now, that's very different than what happens over here, which is called the light-independent reaction, meaning that it doesn't need light. And there's another name for that that we're going to write, which is the Calvin cycle, named after the scientist who kind of solidified our understanding of this process. Okay, these two terms are synonymous. If you're really old like me, then in high school you probably learned them as the dark reactions, but no one says that anymore because that's scary. Okay, so we just call it either the light independent or Calvin cycle. Now, the whole goal of photosynthesis is to create glucose. Okay, so I'm going to go ahead and write that so that I keep that in mind, okay, that I gotta make this. So let's talk about where these are coming from, shall we? The hydrogens are going to come from here, okay? So I'm gonna steal the hydrogens from NADPH, and now I don't have NADPH anymore, I just have NADP, okay, because I'm going to use the hydrogens to make my glucose. All right, now I need C's and I need O's. Oh, well, I know what we need. We need carbon dioxide. Okay, that's the other reactant that we haven't drawn in yet. So that's going to come into play in this light-independent reaction. And now I have all of the things that I need, C's, H's, and O's. Okay, but I have one problem. The bonds in this glucose molecule are super high energy. And in order to form those bonds, I'm going to need energy. That's an anabolic reaction. I need energy to do that. Well, that energy is going to come from our friend ATP. So I'm going to send ATP over here. I'm going to use the energy to form these high energy bonds. Okay, and then I don't have ATP anymore, I have ADP. So don't get fooled, okay? Photosynthesis does produce ATP, but it's not for the rest of the cell to use. It produces it and uses it in the same process, so it's really not available for cells to use. All right, I'm going to do one last thing here, which is to trace the energy conversion. Okay, so again, we're starting off with energy in the form of light. In the light dependent reaction, all of that energy in light, okay, is being transferred to the chemical bonds or electrons in these two molecules. And in the Calvin cycle or light independent reactions, okay, I'm going to use the energy from these two molecules to form the high energy bonds in glucose. Okay, so overall, I'm going from light energy to chemical energy in the bonds of glucose. So that is an example of chemical energy. Okay, so at this point you should be able to fill this in on your own. So why don't you take a moment to pause this, okay, and then start playing it again when you're ready to check your answers. Okay, the light-dependent reaction takes place in the thylakoids. It starts with water, and we also have ADP and NADP, and that's it. Okay, this process is going to release oxygen. We're going to recharge ADP into ATP, and we're going to attach an electron and a hydrogen atom to make NADPH. We are going from light energy to chemical energy, okay, and that chemical energy is here, okay, and you can work on a general description of your own. Use that diagram we just drew. In the light-dependent reaction, that takes place in the stroma, and I said it wrong again, light-independent reaction, gosh darn it, it takes place in the stroma. Okay, we need a couple things for that. We need carbon dioxide, we need that high energy ATP, and we need the NADPH. Throughout that Calvin cycle, we're going to fix that carbon and make it into glucose. Okay, and then we're back to ADP and NADP because we used the energy and electrons from these guys to make this glucose. Okay, so here we're going from chemical energy in these molecules, and we're ending with chemical energy in the bonds of glucose. So overall, this happens in the chloroplast. We start with water and carbon dioxide, and I'm not going to include these molecules because they stay within the chloroplast. Okay, they never leave, they're not really involved in any reactions because they just cycle through. And we're going to end with glucose and oxygen. We're going from light energy to chemical energy in the bonds of glucose overall. And remember, if you're doing these notes from my class, you should be able to fill in these boxes on your own. Okay, so now we're going to take a look at how oxygen is affected over the course of a couple of days with a photosynthetic organism. Okay, so go ahead and work on maybe setting up a graph here. On our x-axis, we've got days and nights over the course of 2 days. And on the y-axis, that looks like it might be kind of hard to see, those are oxygen levels. So we've got an arbitrary zero line here, okay, meaning anything above here represents oxygen production, okay, levels of oxygen going up. and anything below this line represents oxygen consumption, so oxygen levels going down. All right, during the day, plants are going to be photosynthesizing. So if they're photosynthesizing, they should be creating oxygen, and that would mean that my oxygen levels should go up during the day, okay? Now, as the sun starts to set and light is no longer available for photosynthesis, we would expect the oxygen levels to fall. But careful here, okay, during the nighttime, actually all the time, plants are taking, and other photosynthetic things, they're taking this glucose that they're making and they're putting it through the process of cell respiration because they need ATP for things, okay? So the whole reason that they're undergoing photosynthesis is to make glucose so they can then undergo respiration to create ATP. So what we're gonna see is a dip in our oxygen levels during the night, okay? Because they're doing this all the time, they're consuming oxygen all the time, but usually it's just outweighed by how much they're producing, okay? So you can think of it as like money, like they're spending a little bit of money, but they're making a lot, okay? When it's not light outside, all they're doing is spending, they're not making any. And so then we see that trend continuing, okay? When the light comes back, the oxygen levels are going to rise, and when the light goes away, they're going to dip down again. So here are the areas where we're doing photosynthesis and cellular respiration, but down here, this is only cellular respiration. We can't do photosynthesis because there's no light. Okay, so if we look at this formula for photosynthesis, okay, I want to figure out how fast photosynthesis is happening. There are a couple of ways that I can do it. I can either measure how much glucose is being produced, so I should see an increase. I could measure how much oxygen is being produced. I should see an increase in that. I can try to measure how much carbon dioxide is being consumed or how much water is being consumed. Okay, now some of these are a little bit easier to measure than others, but things that photosynthesis uses should go down, things they produce should go up. Now, oxygen is one of those things that should go up. So one of the things that we can do is we can measure oxygen production. This is a lot easier in aquatic plants because we can simply stick them underwater, and as they're producing oxygen, that oxygen is going to be released as oxygen gas, which we're going to see as bubbles. Could we do this with a terrestrial plant? Sure. Okay, we could like stick it in a box and then stick an oxygen sensor, okay, on there to see how this plant's environment is changing in terms of oxygen levels. Okay, but counting bubbles is a good one here for aquatic plants. The bad news is that it needs to be adjusted for cellular respiration. Okay, cellular respiration is being done at all times, okay, because they're taking that glucose that they're making in cell respiration, that they're making in photosynthesis, and they're putting it through cell respiration in the mitochondria in order to generate ATP molecules. They need these guys, so they're going to take what they make, again, glucose, in photosynthesis, put it through respiration for ATP. One of the problems is that process consumes oxygen. So whatever this is making is only the difference between the oxygen that we make in photosynthesis, okay, minus the oxygen that we use in cellular respiration. And so here all we're seeing is a net oxygen production, so some adjustment has to be made. This is what we call a direct measurement because we're measuring a direct product of photosynthesis, the oxygen. The other thing we could do is measure how much carbon dioxide is being absorbed. So photosynthesis requires carbon dioxide, so we should see a decrease in carbon dioxide. Again, there's a couple ways you can do that. You could put your plant in a reaction chamber, hook up a carbon dioxide sensor, and actually measure it. if these kinds of things aren't available to you, one of the things that you can do is measure the pH of the surrounding environment. Again, a lot easier if you're talking about an aquatic plant. Carbon dioxide, when it gets absorbed into water, okay, makes an acid, okay? So when I have a lot of carbon dioxide in there, okay, I'll write this over here, a lot of carbon dioxide equals a drop in pH. Now, as plants absorb that carbon dioxide, okay, the pH should start to go up. Okay, carbon dioxide makes an acid, so if I have less of it, it should be less acidic, okay, meaning I'm getting more towards a neutral pH here. So again, this has to be adjusted for cellular respiration because as plants are absorbing carbon dioxide for photosynthesis, they are also producing it during respiration, so there's a little bit of an adjustment there. Again, this is a direct measurement because it's a direct product of photosynthesis. And the last one we're going to talk about here are changes in biomass. So in other words, like how much did the plant weigh before and after? And so what this kind of shows is how much glucose was produced. So glucose is a product of photosynthesis. Photosynthesis kind of allows for the production of glucose, which then if we have a bunch of it, then we can use that glucose to make structural compounds like cellulose, okay? And all of that is needed to build new cells. So as I'm building new cells, I would also be gaining mass, okay? Since that glucose is used, okay, to add on to that mass there. The problem is it may not be due solely to photosynthesis. There could be other factors there, like is it absorbing water from the environment? Okay, are there some other factors that are going on? Is it having to expend a lot of that energy that it wouldn't normally be doing? And so this is what we call an indirect measurement. We're assuming that one of the products of photosynthesis is making a change in what we're measuring, okay, but we're not measuring an exact product of the process. Okay, now in IB world, okay, there are 6 graphs that you have to know. 3 of them are from this chapter. That's a 3, wow. 3 of them are from our chapter on photosynthesis. The other 3 are from a previous chapter on enzymes. So before we go on, let's quickly review here. So we know there are 3 factors that influence enzyme activity: temperature, pH, and substrate concentration. And they have graphs that pretty much look like this. Okay, we have a linear increase until we reach an optimal temperature, and then we have a denaturation. For enzymes, okay, and pH, we've got denaturation into acidic environment, an optimal pH, and then denaturation in too much of a basic environment. Remember, the optimal temperatures and optimal pHs differ for different enzymes. And then for substrate concentration, okay, we see an increase and then a plateau as every enzyme becomes busy with a substrate. So these are the 3 that I need to know for enzymes. The 3 that I need to know for photosynthesis all have to do with the things that actually affect the rate. So here we're talking carbon dioxide concentration, light intensity, and temperature. Let's do temperature first. Okay, so I'm going to put temperature down here on my x-axis, remembering that every photosynthetic organism is going to have a different optimal temperature. And on my y, this is going to be rate of photosynthesis. Okay, can you please spell that out? I don't have enough room. Okay, so here's our general trend. It's very similar to the one in enzymes. We're going to see a linear increase, some kind of optimal temperature, and then a pretty swift drop off here. So let's label, this is my optimal temperature. Okay, over here, this rate is increasing pretty steadily because increasing the temperature increases. Let's pretend that says increasing. Holy crap. Increasing molecular reactions. Okay, so as the temperature goes up, Okay, things are colliding more often with more energy, so reactions tend to speed up. Okay, and so what happens here on the backside? Well, just like with almost every other metabolic process, photosynthesis is controlled via enzymes. So if we increase the temperature too much, okay, we get a denaturation of enzymes. And just so we don't get confused, we'll add needed for photosynthesis. Okay, so this is not denaturation. This is just things are slow because reactions are slow at cold temperatures. We have an optimal temperature, and out here we've denatured or ruined the active site shapes of all the enzymes we need for photosynthesis to happen. Okay, next thing we're going to take a look at is light intensity. And that can mean a couple of things. Like if we're talking about a lamp, maybe something with more wattage, maybe something where the light source is closer. Okay, so the intensity of the light source. That is going to look something like this. Okay, we're going to see that down here at 0 light, we have a 0 rate of photosynthesis. Photosynthesis cannot occur without light. So that wants to start down there, okay? And then things are going to increase. And that makes sense to me because as we're adding more light, we can do more of that photolysis process, okay? And things can just generally happen faster, okay? And then we're eventually going to see a plateau, okay? So again, up in here, more light energy means more photolysis, okay, and some other things which we'll get into if you're lucky enough to be into higher level, okay, but we see a general increase here, and then we see a point at which, okay, all of the enzymes needed for photosynthesis are already working at max speed. Okay, so adding any more energy in here in the form of light isn't going to be helpful after a certain point. Okay, everything's already going as fast as it can. And now let's take a look at our last factor, okay, carbon dioxide concentration and the rate of photosynthesis. So we know that carbon dioxide is needed for the Calvin cycle, okay, which is our light independent reaction. Okay, so if I don't have any carbon dioxide, I can't produce any glucose. I don't have the carbons that I need. So at 0 carbon dioxide, I've got a rate of 0. Again, as I increase the concentration of carbon dioxide, then all of those areas where the light independent reactions are able to take place, okay, are going to start to have the materials they need in order to make that glucose. So as I increase the concentration, I'm able to perform more of those reactions. Of course, there's a limit to that, okay, so at a certain point, again, I'm going to get a plateau. For the same reason that I got a plateau with light intensity. All of the molecules and enzymes that are necessary for carbon fixation in that Calvin cycle, they're already working at maximum capacity. If I give them any more raw materials, it doesn't make a difference. And that'll do it for chapter 2.9 on photosynthesis.